We are Laura, Biel, Gerard, and Xènia, and in this video we are going to visualize the protein COP transcription factor 2, which gen is NR2F2, present in Homo sapiens. Its UniProt code is P24486, and in order to visualize better our protein, we selected the PDB entry 3CJW, which its resolution is 1.48 angstroms, which grabs the amino acid position 175 to 414. And the method to get the PDB structure is with X-ray.
Protein COP transcription factor 2 is located in the nucleus of the cell, and it is a ligand-activated transcription factor. This protein is activated by high concentrations of 9-cis-rhetoric acid and all-trans-rhetoric acid. Its function is the regulation of the apolipoprotein A1 gene transcription, and it binds to DNA site A. The protein has different regions. DNA binding, that goes from position 76 to 151, and two zinc fingers, the first one going from 79 to 99, and the second one from position 115 to 139. The molecular function of the protein are DNA binding transcription factor activity, RNA polymerase II specific, nuclear receptor activity, protein homodimerization activity, rhetoric acid binding, RNA polymerase II regulatory region sequence-specific DNA binding, sequence-specific DNA binding, steroid hormone receptor activity, transcription co-repressor activity, zinc ion binding.
Protein, as all other proteins, is composed of different amino acids. The secondary structure is composed of 13 alpha helix and loops that change the direction of the polypeptide chain. The supersecondary structure is an alpha hairpin because we can only visualize alpha helices. Tertiary structure. We can see that the protein is formed by two domains forming an orthogonal bundle. This protein, for the quaternary structure, can be either homodimer and heterodimer. On one hand, the protein is homodimer when the two domains are the same. On the other hand, when the domains are different and this domain bonds with the NR2F6 protein, it is considered heterodimer.
In our protein, we have 6 natural variants. In our primal fetch, we are able to see just 4. Of these, 2 are not related to diseases, and the other 2 are. In position 25, there is a natural variant that makes the protein have 2 glutamines instead of 1. This mutation results in reduced transcription activation of the EGR1 promoter. Now we look at the variants that are present in our fetch. First, we look at the ones that are related to disease. In position 205, there is one that changes from an aspergine that is polar to an isoleucine that is apolar. This big change increases the transcription of EGR1 gene. Another natural variant of this protein is in the position 341, colored in red, and changes from a serine to a tyrosine. The mutation results in reduced transcriptional activity. The error of this protein results in a disease called congenital heart defects multiple types 4 that affects the shape of the heart. This disease is genetic and is given from birth. On the other hand, we have the natural variants that don't affect the disease. In position 251, marked in blue, we can have a change from glutamic acid to an aspartic acid. And finally, we have another at position 412 that changes from an alanine to a serine. Even though it is changing from an apolar to a polar amino acid, this change isn't related to any disease.